Assalamualaikum warahmatullahi wabarakatuh. My name is Fatimah Syahada binti Anwarul Haq, matric number 170878808. Stroke 1. I will present to you about the development of transgenic crops. These are the table of contents for my presentation. First, we will look to the transgenic crop that I chose, following with the problems with the chosen crop. Third is the development of transgenic crop. Next is the engineering method. Fifth is the expected benefits. And lastly, references. For transgenic crop, I chose mangoes. The reasons I chose mangoes because mangoes are one of the top choice and well accepted fruits all over the world. Mango ranks as the fifth most consumed fruit in the world after citrus, banana, grapes, and apple. According to Malaysian Gross Domestic Product (GDP), fruits industry contributed an addition of 4.78 billion ringgit in 2019. The Ministry of Agriculture and Food Industries (MAFI) also listed that mango as the 12th major tropical fruits in Malaysia. Malaysia produced 10.5 thousand tons of mangoes in 2018 according to FAO. So what are the problems with mangoes? Mangoes are susceptible to post-harvest diseases such as anthracnose. Anthracnose is a fungicidal infection caused by Coricotrichum cuspidis. Conley et al. 2016 reported that anthracnose disease is the most serious biological constraint to mango production. C. cuspidis is one of the frequently reported plant pathogens among genus Coricotrichum in India, reported by Komari et al. 2017, and the diagram on the right shows the structure of C. cuspidis. Anthracnose turns the whole mango blackish and shrivels until it is no longer suitable for consumption by Prakash et al. 2011. Diagrams below showing the symptoms of anthracnose disease caused by Coricotrichum cuspidis in mango, which are leaves showing bird eye spots, twig, and sunken dark brown black spots that can be seen on mango ripe fruit. Does this post-harvest disease caused by this particular fungicidal attack affect the amount of marketable fruit? Next, we will proceed with the development of transgenic mangoes. So, first we have to isolate one acetoxy 2-hydroxy for oxohemi-icosa 12, 15-dimene, or known as persine. We can isolate the persine by thin layer chromatography from an ethanolic extract partitioned with dichloromethane. Persine is an antifungal diene compound that can be found from unripe fruits. A study in Sri Lanka shows that the resistance of unripe avocado attack by C. cuspidis was due to the presence of the preformed antifungal diene compound, which is persine. Next, we have to prepare a vector to move the DNA of interest into the mango. As we can see here, the vector consists of antifungal diene gene, which is persine, the reporter gene, constitutive promoter, and 1-methyl-cyclopropene. The reporter gene plays a role in to confirm whether the persine gene reached the nucleus of mango cells. And by having constitutive promoter, the antifungal diene gene will be transcribed and its protein will be translated all the time. However, with persine, there is a decline in persine to sublittle once fruit is having ripening, which eventually initiate the development of pathogen. Therefore, 1-methyl-cyclopropene which is an ethylene competitive inhibitor, can interact irreversibly with ethylene receptors, and as a result, a delay in rate of respiration and also ethylene production could occur. Third, we have to introduce the vector into the mango cells using a high insertion device or gene gun. Fourth, we have to grow the plants. And lastly, for the verification of gene expression, we can infect the transgenic mangoes with Collitotrichum glosporidis and observe whether the mango is resistant towards the fungi. Next is engineering method. For this transgenic mango, I chose transformation. Transformation is a process by which a copy of genetic material from a specific trait is inserted into the cells. So in this particular transgenic crop, a high-speed particle bombardment method is used, which assists by a high insertion device. And lastly, for expected benefits, transgenic mangoes are able to produce fungi toxic compound and become resistant towards Pseudosporides infection. And transgenic mangoes are also able to support small farmers' income as it can prevent from devastation caused by the fungicidal infection. A new variant of mangoes can also be produced. So these are the references, and thank you for listening.